Unveiling the mysteries of inheritance. Have you ever wondered why your eyes are the same color as your dad's, or why your friend can roll their tongue while you can't? These are just a few examples of the fascinating world of genetics, the branch of biology that explores how traits are passed down from parents to offspring. It's a captivating story that unfolds like a detective mystery, piecing together clues to understand how living organisms inherit and display the characteristics we see. Before diving into the nitty-gritty of genes and alleles, let's set the stage. Imagine a family photo album, but instead of pictures, it holds the blueprints for every living thing. These blueprints, encoded in a molecule called DNA, deoxyribonucleic acid, contain the instructions that determine everything from eye color to hair texture, and even our susceptibility to certain diseases. Our story begins with the concept of inheritance. It's the remarkable ability of parents to pass on some of their traits to their children. Think about the similarities you share with your parents or siblings. These are inherited traits. But here's the twist. Not all traits are inherited in the same way. Sometimes, a child inherits a trait that neither parent appears to have. How is this possible? This is where variation comes in. Variation refers to the differences we see among individuals within a species. It's what makes each of us unique, even within families. Variation is crucial for the survival of species because it allows populations to adapt to changing environments. Imagine if everyone in a population looked and behaved exactly the same. If a new disease emerged, it could potentially wipe out the entire population. Variation, on the other hand, provides a buffer. Some individuals might have a genetic makeup that makes them resistant to the disease, allowing them to survive and pass on their genes to future generations. Understanding how these two concepts, inheritance and variation, work together is the essence of genetics. But the story wouldn't be complete without a key figure, Gregor Mendel. Gregor Mendel, the father of genetics. In the mid-1800s, an Austrian monk named Gregor Mendel embarked on a series of experiments that would revolutionize our understanding of heredity. Unlike many scientists of his time who focused on studying complex traits in animals, Mendel chose to work with pea plants. These plants have several distinct traits, like flower color, purple or white, and seed shape, round or wrinkled. They also reproduce quickly, allowing Mendel to observe multiple generations in a relatively short time frame. Mendel's meticulous experiments involved carefully controlled crosses between pea plants with different traits. He meticulously tracked the results of these crosses, recording the appearance of the offspring, called the F1 generation, and their offspring in turn, the F2 generation. Through his work, Mendel discovered fundamental principles that govern how traits are inherited, paving the way for modern genetics. The chapter ahead. This chapter will be your guide as we delve deeper into the captivating world of genetics. We'll begin by unraveling the secrets Mendel uncovered, exploring his groundbreaking laws of segregation and independent assortment. You'll learn about the concept of alleles, the different forms a gene can take, and how they determine dominant and recessive traits. We'll also master the art of using Punnett squares, a powerful tool for predicting the probability of different trait combinations in offspring. But genetics isn't just about the simple inheritance patterns observed by Mendel. We'll venture beyond these foundations to explore the fascinating world of genetic diversity. This diversity arises from various factors, including mutations in DNA, the incredible process of genetic recombination during meiosis, and the flow of genes within populations. The journey doesn't end there. We'll delve into the fascinating realm of sex-linked traits, which exhibit unique inheritance patterns because of their location on the sex chromosomes. Pedigree analysis, a tool for tracing trait inheritance across generations, will also be introduced to help us visualize these patterns. By the end of this chapter, you'll have a solid foundation in the basic principles of genetics. This understanding will pave the way for future chapters where we'll explore the intricate world of DNA, protein synthesis, and how genetic information shapes not only individual traits, but also the evolution of species over time. So, buckle up and get ready to embark on this exciting adventure into the world of genetics. The foundations of genetics, unveiling Mendel's legacy. Before the dawn of modern genetics, the mysteries of inheritance remained largely unexplained. Then came Gregor Mendel, an Austrian monk whose meticulous experiments with pea plants in the mid-1800s laid the groundwork for our understanding of how traits are passed from one generation to the next. Mendel's mastermind, the power of pea plants. Mendel wasn't the first scientist to study heredity. However, his genius lay in his strategic choice of a model organism, the pea plant, Pisum sativum. Pea plants possess several key characteristics that made them ideal for genetic research. Distinct traits. Pea plants exhibit easily observable traits like flower color, purple or white, and seed shape, round or wrinkled. These traits have clear-cut categories, making it easier to track inheritance patterns. Self-pollination versus cross-pollination. Pea plants can normally self-pollinate, meaning the pollen from a flower fertilizes the ovules of the same plant. However, Mendel could also artificially cross-pollinate them by transferring pollen between different plants. This allowed him to control which traits were passed on to the offspring. Short generation time. Pea plants have a relatively short generation time, allowing Mendel to observe multiple generations in a controlled time frame. This was crucial for identifying patterns in inheritance. The experiments begin, unpacking segregation. Mendel's experiments focused on analyzing the inheritance of a single trait at a time. He started with pure-breeding pea plant lines, which means they consistently produced offspring with the same trait for generations. For example, a pure-breeding line with purple flowers would always produce offspring with purple flowers when self-pollinated. Mendel then conducted a now-famous cross between a pure-breeding line with purple flowers, PP, and a pure-breeding line with white flowers, WW. This is called AP generation, parental generation. The resulting offspring, the F1 generation, first filial generation, surprised him. All the F1 plants had purple flowers. This observation led Mendel to propose his first law of heredity, the law of segregation. This law states that each individual carries two alleles, or alternative versions, of a gene for a particular trait. In Mendel's experiment, the purple flower allele, P, was dominant over the white flower allele, W. In the F1 generation, the dominant purple allele masked the recessive white allele. Independent assortment, unveiling the mix. Mendel didn't stop there. He allowed the F1 plants, all purple-flowered, to self-pollinate. The resulting offspring, the F2 generation, second filial generation, displayed a surprising ratio of 3:1. 3/4, 75%, of the F2 plants had purple flowers and the remaining 1/4, 25%, had white flowers. This observation led to Mendel's second law of heredity, the law of independent assortment. This law states that genes for different traits are inherited independently of each other during the formation of gametes, sperm and egg cells. During meiosis, the process that creates gametes, the two alleles for each gene segregate and end up in separate gametes. This means that the inheritance of one trait, like flower color, does not influence the inheritance of another trait, like seed shape. Mendel's laws of segregation and independent assortment provided a powerful framework for understanding how traits are inherited. These fundamental principles laid the foundation for modern genetics, paving the way for further research into more complex inheritance patterns. Beyond Mendel, a world of complexity. Mendel's work focused on simple traits with clear-cut dominant and recessive alleles. However, the world of genetics is not always so straightforward. In the following chapters, we'll explore some exceptions and complexities that go beyond Mendelian inheritance. Incomplete dominance. In some cases, neither allele is completely dominant. The heterozygous individual displays an intermediate phenotype, a blend of the two parental traits. Codominance. Here, both alleles are fully expressed, leading to a combined phenotype in the offspring. Polygenic traits. Many traits, such as human height or eye color, are influenced by multiple genes, each contributing a small effect. The environment can also play a role in shaping these traits. By understanding Mendel's principles as a foundation, we can delve deeper into the fascinating complexities of genetics, unlocking the secrets of inheritance and variation in the living world. 3. Unveiling the odds, Punnett squares, and predicting inheritance. Imagine flipping a coin. There's a 50% chance it will land on heads and a 50% chance it will land on tails. Predicting the outcome of a single coin toss might seem simple, but what if you wanted to predict the outcome of flipping multiple coins at once? Genetics, like coin flips, involves probabilities, and a powerful tool called a Punnett square helps us navigate these probabilities when predicting inheritance patterns. The birth of the Punnett square. Developed by Reginald Punnett, a British geneticist, the Punnett square is a visual tool that allows us to predict the possible genotypes, genetic makeup, and phenotypes, observable traits, of offspring resulting from a particular cross between parents. Building the square. Let's use Mendel's classic experiment with pea flower color, purple or white, as an example. Remember, each pea plant carries two alleles for flower color, either a dominant purple allele, P, or a recessive white allele, W. Draw a grid. Create a grid with 2 rows and 2 columns. Label the axes. Along the top row, list the possible alleles the male parent can contribute to his sperm cells. Since pea plants are diploid, having 2 copies of each chromosome, this parent could have either PP, both dominant alleles, or PW, one dominant and one recessive allele. Therefore, label the top row with P and PW. List female alleles. Along the left-hand side column, list the possible alleles the female parent can contribute to her egg cells. Label this column with P and PW as well. Filling the grid. Probability at play. Now comes the magic. Each square in the Punnett square represents a possible combination of alleles an offspring can inherit from both parents. To fill the squares, consider that during gamete formation, meiosis, the 2 alleles for flower color segregate and end up in separate sperm and egg cells. Filling the top row. Since the male parent with genotype PP can only produce sperm cells with the dominant P allele, fill both squares in the top row with P. Filling the left column. Similarly, the female parent with genotype PP can only produce egg cells with the P allele. Fill both squares in the left column with P. Unveiling the cast, dominant, recessive, and the spectrum in between. Imagine a stage play where some actors always get the starring role, dominant alleles, while others are relegated to the background, recessive alleles. This analogy captures the essence of dominant and recessive traits, 2 fundamental concepts in Mendelian genetics introduced in the previous sections. But the world of inheritance isn't always a clear-cut case of leading roles and understudies. As we delve deeper, we'll discover a fascinating spectrum of interactions between alleles that paint a more nuanced picture. Dominant and recessive, the classic duo. Our journey begins with the familiar duo, dominant and recessive alleles. Remember, alleles are different versions of a gene that can determine an organism's traits. In Mendelian inheritance, when an organism inherits 2 different alleles for a trait, one allele, dominant, will usually mask the effect of the other allele, recessive, in the organism's phenotype, observable trait. Think back to Mendel's pea plant experiments. The purple flower allele, P, was dominant over the white flower allele, W. An organism with 2 dominant alleles, PP, or one dominant and one recessive allele, PW, will have purple flowers. Only when an organism inherits two recessive alleles, WW, will the white flower trait be expressed. Dominance, a spectrum, not a binary switch. While dominant and recessive seem like clear-cut categories, the reality can be more nuanced. Dominance can sometimes be incomplete, meaning neither allele is completely dominant over the other. In incomplete dominance, the heterozygous individual, with one dominant and one recessive allele, displays an intermediate phenotype, a blend of the two parental traits. For example, in some Snapdragon varieties, the homozygous dominant, RR, genotype results in red flowers, while the homozygous recessive, RR, genotype results in white flowers. When a red snapdragon, RR, is crossed with a white snapdragon, RR, the F1 generation, RR, has pink flowers, showcasing an intermediate phenotype due to incomplete dominance. Codominance, sharing the spotlight. In some cases, both alleles find a way to share the stage. This is called codominance. Here, both the dominant and recessive alleles are fully expressed in the offspring, resulting in a combined phenotype. A classic example is the ABO blood group system in humans. There are three main alleles for blood type, A, B, and O. Individuals with genotype AA have type A blood, those with BB have type B blood, and those with OO have type O blood. However, individuals with genotype AB have both the A and B alleles fully expressed, resulting in type AB blood, where both A and B antigens are present on red blood cells. Beyond simple Mendelian inheritance, polygenic traits and the environment's influence. Dominant and recessive inheritance provide a powerful framework for understanding basic inheritance patterns. However, many traits in nature, particularly in complex organisms like humans, aren't controlled by single genes with clear-cut dominant and recessive effects. These traits, called polygenic traits, are influenced by multiple genes, each contributing a small effect. For example, human height, eye color, and skin color are all polygenic traits. The environment also plays a role in shaping phenotypes. Imagine a plant with the genes for tall growth, dominant. However, if the plant doesn't receive enough sunlight or nutrients, it might not reach its full potential height. This interplay between genes and the environment is crucial for understanding how organisms develop and adapt to their surroundings. The power of combining tools, Punnett squares and beyond. Punnett squares are a valuable tool for predicting inheritance patterns for traits controlled by single genes with dominant and recessive alleles. However, when dealing with polygenic traits or complex inheritance patterns, Punnett squares become less effective. Modern genetic analysis tools such as DNA sequencing and pedigree analysis help us explore these complexities. Pedigree analysis, for instance, allows us to trace the inheritance of traits through generations in a family, revealing patterns that might not be readily apparent with Punnett squares alone. The expanding stage, genes, environment, and the intricate dance of inheritance. The world of inheritance is far more nuanced than a simple dominant and recessive drama. Incomplete dominance, codominance, polygenic traits, and environmental influences all play a role in shaping the diverse tapestry of life. As we move forward, we'll explore the intricate dance between genes and environment, delve into the fascinating world of sex-linked traits, and unlock the secrets hidden within our DNA. By understanding these complexities, we gain a deeper appreciation for the remarkable mechanisms that govern how traits are inherited and how variation arises within populations. Beyond the simple, sex-linked traits, pedigree analysis, and unveiling genetic diversity. Our exploration of genetics has unraveled the fascinating concepts of dominant and recessive alleles, Punnett squares, and the complexities of polygenic traits. Now we delve deeper, venturing into the realm of sex-linked traits and pedigree analysis and uncovering the fascinating world of genetic diversity that goes beyond simple Mendelian patterns. Sex on the chromosomal stage, unveiling sex-linked traits. Our chromosomes, the tightly packed bundles of DNA within our cells, hold the blueprints for our traits. However, there's a twist when it comes to sex chromosomes. Humans, like many other animals, have two sex chromosomes, X and Y. Females typically have two X chromosomes, XX, while males have one X and one Y chromosome, XY. This difference in sex chromosomes plays a crucial role in determining the inheritance patterns of certain traits, known as sex-linked traits. One of the sex chromosomes, the X chromosome, carries many genes that influence various traits, not just those related to sex determination. The Y chromosome, on the other hand, is much smaller and carries fewer genes. This difference in gene content creates unique inheritance patterns for sex-linked traits. A spotlight on X-linked traits. The most common type of sex-linked trait is the X-linked trait. These traits are determined by genes located on the X chromosome. Since males have only one X chromosome, any recessive allele they inherit for an X-linked trait will be expressed in their phenotype. This explains why some X-linked recessive disorders, like red-green colorblindness, are much more common in males than in females. Females, with two X chromosomes, have a higher chance of escaping the effects of a recessive X-linked trait. If one X chromosome carries the dominant allele, it can mask the effect of a recessive allele on the other X chromosome. However, females can still be carriers of an X-linked recessive trait, meaning they don't show the trait themselves but can pass the recessive allele on to their offspring. Unlocking family histories. Pedigree analysis. Imagine a visual representation of a family tree, but instead of names, it charts the inheritance of traits across generations. This is the essence of pedigree analysis. This valuable tool allows us to track the inheritance patterns of traits, including sex-linked traits, within a family. Pedigree symbols represent individuals, squares for males, circles for females, and lines connect them to show parents and offspring. Shaded symbols indicate individuals who express a particular trait. By analyzing the patterns of inheritance shown in a pedigree, geneticists can make inferences about the underlying genes and their dominance patterns. Beyond simple Mendelian patterns, unveiling genetic diversity. Mendelian inheritance provides a foundation for understanding basic inheritance patterns. However, the world of genetics is far more diverse. Let's explore some factors that contribute to this rich tapestry of variation. Mutations. Mutations are permanent changes in an organism's DNA sequence. These changes can occur spontaneously during cell division or be caused by environmental factors like radiation. Mutations can have various effects. Some might be neutral, while others can create new traits or lead to diseases. Genetic recombination. During meiosis, the process that creates sex cells like sperm and egg, a fascinating phenomenon called genetic recombination occurs. Here, homologous chromosomes, chromosome pairs, exchange genetic material through a process called crossing over. This reshuffles the alleles on chromosomes, creating new combinations in offspring. Genetic recombination plays a crucial role in generating genetic diversity within populations. Gene flow. The movement of genes between populations is known as gene flow. This can happen through migration or interbreeding between populations. Gene flow introduces new alleles into a population, increasing variation and allowing populations to adapt to changing environments. The power of diversity, adaptation, and evolution. Genetic diversity within a population is crucial for survival and adaptation. Imagine a population of organisms with all the same genes. If a new disease emerges, it could potentially wipe out the entire population. However, the presence of genetic variation allows some individuals to have traits that make them resistant to the disease. These individuals survive and reproduce, passing on their resistant genes to future generations, allowing the population to adapt and thrive. Genetic diversity is not only essential for adaptation within populations, it's also the driving force behind evolution. Over vast timescales, changes in gene frequencies across populations lead to the emergence of new species through the process of natural selection. Looking ahead, from simple traits to the complexities of DNA, we've begun to peel back the layers of the fascinating world of genetics. We've explored basic inheritance patterns, unraveled the intricacies of sex-linked traits, and peeked into the world of genetic diversity, unveiling the engine of inheritance, mutations, and the dance of DNA. Our journey through genetics has revealed the captivating world of inheritance, from the classic principles of dominant and recessive traits to the complexities of sex-linked inheritance. Now we delve deeper, exploring the engine that drives this remarkable process: mutations in DNA, the molecule that holds the blueprints for life. DNA, the blueprint of life. Imagine a library containing all the instructions needed to build and maintain an organism. This library is DNA, deoxyribonucleic acid, a complex molecule that resides within the nucleus of every cell. DNA is a double-stranded molecule shaped like a twisted ladder, with the rungs formed by pairs of chemical units called nucleotides. The sequence of these nucleotides, like letters in a code, determines the genetic information passed from parents to offspring. Mutations, the spark of change. Mutations are permanent changes in the DNA sequence. These changes can be caused by various factors, including errors during DNA replication, exposure to environmental mutagens like radiation, or even random events. Mutations can affect single nucleotides or larger segments of DNA. The spectrum of mutations. Mutations can have a wide range of effects on an organism. Silent mutations. Some mutations might not cause any noticeable change in the organism's phenotype, observable trait. This is because the change in DNA sequence might not alter the amino acid sequence of the protein coded by that gene. Beneficial mutations. In some cases, mutations can be beneficial. For example, a mutation might create a new protein variant that provides an organism with an advantage in its environment, such as increased resistance to disease. Detrimental mutations. Some mutations can be detrimental, leading to malfunctions in proteins or even cell death. These mutations can cause genetic disorders or diseases. The delicate dance of DNA repair. Cells have built-in mechanisms to detect and repair mutations in DNA. These repair mechanisms help maintain the integrity of the genetic code and ensure its faithful transmission to offspring. However, sometimes repairs are not successful and the mutation persists, potentially impacting the organism. The domino effect of mutations. Mutations can have a ripple effect, influencing the proteins encoded by the affected gene. Proteins are the workhorses of the cell, carrying out various functions essential for life. If a mutation alters the structure or function of a protein, it can disrupt cellular processes and lead to abnormal development or disease. The evolutionary significance of mutations. Mutations are the raw material for evolution. By introducing changes in the genetic code, they create variation within populations. This variation provides a pool of possibilities for natural selection to act upon. Organisms with beneficial mutations are more likely to survive and reproduce, passing on those mutations to future generations. Over vast timescales, this process of accumulating mutations and natural selection can lead to the emergence of new species. Beyond simple mutations, exploring other sources of variation. While mutations are a crucial source of genetic variation, other factors contribute to the rich tapestry of life. Genetic recombination. During meiosis, homologous chromosomes, chromosome pairs, exchange genetic material. This reshuffles the alleles within chromosomes, creating new combinations in offspring and increasing variation. Gene duplication. Sometimes, sections of DNA are duplicated during cell division. These duplicated genes can evolve new functions, contributing to the complexity of organisms. Horizontal gene transfer. In some cases, genetic material can be transferred between organisms that are not parent and offspring. This can happen through viruses or bacteria, introducing new genes into an organism's genome and increasing variation. The importance of genetic diversity. Genetic diversity within a population is crucial for survival and adaptation. A population with a wider range of genetic variations is better equipped to handle environmental changes. For example, if a new disease emerges, some individuals might have mutations that confer resistance. This allows the population to adapt and survive. Looking ahead, from mutations to the power of DNA technology, we've explored the fascinating world of mutations and their role in creating genetic variation. In the following chapters, we'll delve deeper into the intricate world of DNA. We'll unravel the process of DNA replication, explore how DNA codes for proteins, and discover the power of modern DNA technology to diagnose diseases, understand human evolution, and even manipulate genes. Decoding the master code, DNA replication, transcription, and the journey from genes to proteins. Our exploration of genetics has unveiled the captivating dance between DNA and inheritance. We've delved into the power of mutations and the importance of genetic diversity. Now, we embark on a fascinating journey to understand how the information encoded within DNA directs the construction of proteins, the workhorses of the cell. This journey involves two crucial processes: DNA replication and protein synthesis, often referred to as the central dogma of molecular biology. DNA replication, the blueprint unfurls. Before a cell can divide, it must ensure that each daughter cell receives a complete copy of the DNA. This remarkable feat is achieved through a process called DNA replication. Imagine a zipper being carefully unzipped. This is what happens to the DNA double helix during replication. Enzymes break the hydrogen bonds between the paired nucleotides, separating the two strands. Each strand then acts as a template for the synthesis of a new complementary strand. New nucleotides are brought in and paired with the exposed bases on the template strand, following the rule of A pairing with T and C pairing with G. The end result is two identical double-stranded DNA molecules, ensuring each new cell inherits a complete copy of the genetic information. Transcription. From DNA to RNA, the message is delivered. DNA is safely tucked away within the nucleus, but how does the information encoded within it reach the ribosomes in the cytoplasm, where protein synthesis occurs? This critical role is played by messenger RNA, mRNA. During transcription, a specific segment of DNA serves as a template for the synthesis of a short, single-stranded mRNA molecule. RNA is similar to DNA, but it contains the sugar ribose instead of deoxyribose and the uracil, U, nucleotide instead of thymine, T. RNA polymerase, an enzyme, moves along the DNA template, guiding the assembly of RNA nucleotides complementary to the DNA sequence. The newly synthesized mRNA molecule, carrying a specific genetic message, then detaches from the DNA and migrates to the cytoplasm, acting as the intermediary between DNA and protein synthesis. The genetic code, breaking the code of life. Imagine a recipe written in a foreign language. DNA is like this recipe, containing instructions for building proteins. But how do cells translate this code? The key lies in the genetic code. This code is a set of instructions that dictates how the sequence of nucleotides in mRNA is translated into a sequence of amino acids, the building blocks of proteins. Each codon, a triplet of nucleotides, 3 nucleotides long, on the mRNA molecule specifies a particular amino acid. For example, the codon August codes for the amino acid methionine, while UU codes for phenylalanine. There are 64 possible codons, and each codes for a specific amino acid, except for 3 codons that signal the end of protein synthesis. Translation: from message to protein, building the cellular workhorses. Transfer RNA, tRNA, molecules are the key players in translation. Each tRNA molecule has 2 crucial features: an anticodon, a triplet of nucleotides complementary to a specific codon on mRNA, and an attachment site for a specific amino acid. During translation, ribosomes, complex structures in the cytoplasm made of RNA and protein, bind to the mRNA molecule. tRNA molecules, each carrying a specific amino acid, line up along the mRNA molecule, their anticodons interacting with the codons on mRNA. Following the genetic code, ribosomes facilitate the formation of peptide bonds between amino acids, linking them into a polypeptide chain, the primary structure of a protein. From blueprint to function, proteins shape the cell. The sequence of amino acids in a protein determines its three-dimensional structure, which in turn dictates its function. Proteins play a myriad of roles within cells, acting as enzymes that drive metabolic reactions, structural components like collagen in bones, or transport molecules that shuttle substances across membranes. Mutations in DNA that alter the sequence of amino acids can disrupt protein structure and function, potentially leading to diseases. Regulation of gene expression. Not all genes are created equal. Our cells contain a vast amount of genetic information, but not all genes are expressed all the time. Gene expression refers to the process by which the information encoded in a gene is converted into a functional product, like a protein. Cells have elaborate mechanisms to regulate gene expression, ensuring that only the genes required for a specific function are expressed at a particular time and in a particular location. This intricate control system allows cells to optimize resource allocation and respond to environmental cues. The future of genetics. Unlocking the potential. Understanding the processes of DNA replication, transcription, and translation has revolutionized our understanding of life. Modern DNA technology allows us to analyze genes, diagnose diseases with greater accuracy, and even develop targeted therapies. Unveiling the human story, DNA technology, and the power of genomics. Our exploration of genetics has taken us on a remarkable journey, from the fundamental principles of inheritance to the intricate dance between DNA and protein synthesis. Now, we delve into the exciting realm of DNA technology and genomics, unlocking the secrets hidden within our own DNA and revolutionizing our understanding of human health, disease, and evolution. DNA fingerprinting, unveiling identity. Imagine a unique identifier as personal as a fingerprint, but based on your DNA. This is the essence of DNA fingerprinting. This technology allows scientists to analyze specific regions of an individual's DNA that are highly variable between people. The resulting pattern, unique to each individual except for identical twins, can be used for various purposes, including forensic science. DNA fingerprinting is a powerful tool in forensic science for identifying suspects or exonerating the wrongly accused. By comparing DNA samples from crime scenes to those of suspects, investigators can establish or exclude links between individuals and evidence. Paternity testing. DNA fingerprinting can be used to determine biological parentage with high accuracy. By analyzing the DNA of a child, mother, and alleged father, scientists can compare genetic patterns to confirm or disprove paternity. Beyond fingerprints, diving deeper with DNA sequencing. DNA fingerprinting provides valuable information, but it only analyzes specific regions of DNA. For a more comprehensive picture, scientists use DNA sequencing technologies. These techniques allow us to determine the exact order of nucleotides, A, C, G, and T, across an entire DNA molecule. The human genome, for instance, contains billions of base pairs. Modern sequencing technologies can rapidly and accurately read these vast amounts of data, revealing an individual's complete genetic makeup. DNA sequencing has revolutionized various fields. Genetic disease diagnosis. By comparing an individual's DNA sequence to known disease-causing mutations, doctors can diagnose genetic disorders with greater accuracy. This allows for early intervention and improved patient management. Personalized medicine. DNA sequencing can help tailor medical treatments to an individual's unique genetic makeup. By understanding how a person's genes might influence their response to medications or their risk of developing certain diseases, doctors can create personalized treatment plans. The age of genomics. Unveiling the human story. The field of genomics focuses on the analysis and manipulation of entire genomes. By studying the collective set of genes within an organism, scientists can gain insights into human evolution. By comparing the genomes of humans and other primates, scientists can reconstruct our evolutionary history and understand how our species came to be. Genetic diversity. Genomic analysis reveals the remarkable diversity within the human population. Studying this variation helps us understand human history, migration patterns, and susceptibility to diseases. The ethical considerations. Balancing progress with responsibility. The power of DNA technology is immense, but with great power comes great responsibility. There are important ethical considerations surrounding the use of genetic information. Privacy. Protecting an individual's genetic privacy is crucial. DNA can reveal sensitive information about an individual's health risks or ancestry, and it's essential to ensure this information is used responsibly and with consent. Discrimination. Genetic information should not be used to discriminate against individuals when it comes to employment, insurance, or access to healthcare. Regulations are needed to prevent such misuse. The future of genomics, a brighter, more personalized tomorrow. The field of genomics is rapidly evolving, and its applications continue to expand. Here's a glimpse into what the future might hold. Gene therapy. Gene therapy aims to correct defective genes that cause diseases. By introducing healthy copies of genes or silencing faulty ones, scientists hope to develop cures for various genetic disorders. Pharmacogenomics. This field focuses on how an individual's genetic makeup influences their response to medications. By understanding these interactions, scientists can develop personalized drug therapies that are more effective and have fewer side effects. The power of genetics, shaping our understanding of life. Our exploration of genetics has unveiled a captivating world where the information encoded within DNA governs the diversity of life, from the inheritance of traits to the intricate dance of protein synthesis. DNA technology has revolutionized our understanding of ourselves and holds immense potential for improving human health and well-being. As we continue to explore the complexities of the human genome, we move towards a future where personalized medicine and gene-based therapies can offer a brighter and healthier tomorrow. Beyond the human, exploring the genetic landscape of our planet. Our genetic odyssey has thus far focused on the fascinating world within us. But genetics isn't just about humans, it's the story of life on Earth, a story written in the language of DNA. In this section, we'll delve into the captivating realm of comparative genomics, exploring the genetic diversity of our planet and its role in evolution, conservation, and agriculture. A tapestry of life, unveiling genetic diversity. Life on Earth is a breathtaking tapestry woven from countless species, each with a unique genetic code. This remarkable genetic diversity is the foundation for the rich biodiversity of our planet. Comparative genomics allows us to compare the genomes of different species, revealing insights into their evolutionary relationships, adaptations, and unique traits. For example, by comparing the genomes of humans and chimpanzees, our closest living relatives, we can see that we share over 98% of our DNA. This shared genetic heritage reflects our common ancestry. However, the remaining 2% difference in DNA sequence accounts for the subtle yet significant differences between our species, such as brain size and cognitive abilities. Evolution's engine, natural selection, and genetic variation. Genetic diversity is the fuel for evolution. Remember, mutations introduce changes in the DNA sequence, creating variation within populations. These variations can influence an organism's traits, such as resistance to disease or tolerance for harsh environments. Natural selection, the driving force of evolution, acts upon this variation. Organisms with traits better suited to their environment are more likely to survive and reproduce, passing on their advantageous genes to future generations. Over vast timescales, this process leads to the gradual adaptation of populations and the emergence of new species. Comparative genomics and conservation, protecting our precious biodiversity. The loss of biodiversity poses a significant threat to the health of our planet. Comparative genomics plays a crucial role in conservation efforts by helping us understand the genetic makeup of endangered species. By analyzing genetic variation within populations, scientists can identify populations at risk due to inbreeding or habitat fragmentation. This information can then be used to design conservation strategies that promote genetic diversity and ensure the long-term survival of these species. The future of food, harnessing genetics for sustainable agriculture. The world's population is growing, and so is the demand for food. Comparative genomics is transforming agriculture by allowing us to understand the genetic makeup of crop plants and livestock. By identifying genes that influence desirable traits like drought resistance, increased yield, or improved nutritional content, scientists can develop new crop varieties and breeding programs that are more productive and sustainable. The ethical considerations, balancing progress with environmental protection. As with any powerful technology, ethical considerations surround the use of comparative genomics in agriculture. Genetically modified organisms, GMOs, have sparked debate about potential environmental risks and the unintended consequences of altering the genetic makeup of food sources. Open dialogue and responsible research are crucial to ensure that advancements in this field benefit both society and the environment. One world, one connected genome, unveiling our interdependence. Our exploration of comparative genomics underscores the interconnectedness of life on Earth. All living organisms share a common genetic ancestry, and the diversity of life is a testament to the power of mutations and natural selection. Understanding the genetic landscape of our planet allows us to appreciate the delicate balance of ecosystems and the importance of preserving biodiversity. The future of exploration, unveiling the microbiome and beyond. The story of genetics extends beyond visible organisms. The human body, for instance, harbors a vast community of microscopic organisms collectively known as the microbiome. These bacteria, fungi, and archaea play a crucial role in our health and well-being. By studying the microbiome through genetic analysis, scientists are unraveling its complex interactions with the human body and its potential role in various diseases. A tapestry woven in DNA. Our journey through genetics has revealed the captivating story of life, a story written in the language of DNA. From the intricate dance of DNA replication to the vast tapestry of genetic diversity across our planet, genetics shapes the living world in profound ways. As we continue to unravel the secrets hidden within DNA, we gain a deeper appreciation for the interconnectedness of life and the power of this remarkable molecule to shape our future. The expanding horizon, frontiers in genetics. Our exploration of genetics has unveiled a fascinating world, from the basic principles of inheritance to the cutting-edge applications of DNA technology. As we stand at the precipice of exciting discoveries, this section delves into the frontiers of genetics, where researchers are pushing the boundaries of our understanding and paving the way for a future filled with possibilities. Gene editing, rewriting the code of life. One of the most revolutionary advancements in genetics is gene editing. Techniques like CRISPR-Cas9 allow scientists to precisely modify an organism's DNA sequence. This has opened doors for potential treatments of genetic diseases by correcting faulty genes or introducing healthy copies. Imagine a future where diseases like cystic fibrosis or sickle cell anemia become a thing of the past, thanks to the ability to edit the underlying genetic code. However, gene editing raises significant ethical concerns. Altering the human germline, sperm and egg cells, could have unintended consequences for future generations. Open discussions and clear guidelines are essential to ensure responsible use of this powerful technology. Personalized medicine, tailoring treatments to your unique DNA. The promise of personalized medicine is becoming a reality. By analyzing an individual's genome, doctors can tailor treatment plans to their specific genetic makeup. This approach considers factors like how a person's genes might influence their response to medications or their susceptibility to certain diseases. Imagine a future where cancer treatments target the specific mutations driving tumor growth, leading to more effective therapies with fewer side effects. Genetic ancestry and identity, unveiling our past. DNA analysis is revolutionizing how we understand our ancestry. Commercial genetic testing services allow individuals to trace their roots back to historical migrations and identify regions where their ancestors originated. This newfound knowledge can provide a deeper sense of personal identity and connection to the past. However, it's important to remember that these tests only capture a portion of our genetic heritage and may not tell the whole story. Synthetic biology, building life from scratch. Synthetic biology pushes the boundaries of what's possible by engineering new biological systems. Scientists can design and create novel DNA sequences or even entire genomes, paving the way for the development of new organisms with specific functionalities. Imagine microbes engineered to clean up pollution or produce biofuels, or even the potential for creating new organs for transplantation. The possibilities are vast, but careful consideration of the ecological and ethical implications is crucial. The future of genetics, a world of opportunity and responsibility. The future of genetics holds immense promise for improving human health, revolutionizing agriculture, and fostering a deeper understanding of life itself. However, with every advancement comes the responsibility to ensure its ethical and responsible use. Open communication, collaboration between scientists and ethicists, and clear regulations are vital to navigate the complexities of this rapidly evolving field. As we stand on the threshold of a new era in genetics, one thing is certain: the journey of discovery has only just begun. The language of DNA continues to unfold its secrets, and the potential applications of this knowledge are boundless. The choices we make today will determine how we harness the power of genetics to shape a brighter future for generations to come. Beyond the textbook, exploring genetics through research and careers. Our exploration of genetics has unveiled a captivating world, but the journey doesn't end here. The field of genetics offers exciting opportunities for those curious about the inner workings of life. This section delves into the world of genetics research and explores potential career paths for those who want to contribute to this dynamic field. The thrill of discovery, unveiling the secrets of DNA. Genetics research is a continuous quest to unravel the mysteries of DNA. Researchers explore various aspects, including gene function, understanding how genes influence traits, development, and disease susceptibility. Genetic regulation, deciphering the intricate mechanisms that control gene expression. Genomics and evolution, studying the collective set of genes, genomes, to understand how organisms evolve and adapt. Gene therapy, developing strategies to correct or replace faulty genes to treat genetic diseases. A day in the life of a geneticist, unveiling the diversity. The work of a geneticist can vary depending on the specific area of research. Here's a glimpse into what a typical day might involve. Lab work, extracting and analyzing DNA samples, conducting experiments like PCR, polymerase chain reaction, or DNA sequencing. Data analysis, interpreting complex data generated from experiments using bioinformatics tools. Scientific writing, communicating research findings through scientific papers and presentations. Collaboration, working with colleagues from diverse backgrounds, like biochemists, computational biologists, and medical professionals. Beyond the lab, diverse careers in genetics. The field of genetics offers a broad spectrum of career paths beyond traditional research. Here are some exciting possibilities. Genetic counselor, providing information and support to individuals and families at risk of genetic disorders. Forensic scientist, using DNA analysis to identify suspects and solve crimes. Bioethicist, examining the ethical implications of new genetic technologies. Science policy advisor, influencing science policy decisions related to genetics and biotechnology. Science communicator, engaging the public with the fascinating world of genetics through writing, education, or media. Equipping yourself for the journey, building a foundation for success. A strong foundation in science, particularly biology and chemistry, is essential for a career in genetics. Developing critical thinking, problem-solving, and analytical skills will equip you to navigate complex scientific concepts. Additionally, honing your communication skills will allow you to effectively share your findings with others. Exploring the landscape, resources for the aspiring geneticist. Numerous resources are available to help you delve deeper into genetics. Online courses, massive open online courses, MOOCs, from universities and platforms like Coursera and edX offer introductory and advanced courses in genetics. Scientific journals and articles, staying updated with the latest research findings by reading reputable scientific journals like Nature Genetics or Science Advances. Research labs and internships, seeking research opportunities at universities or research institutions can provide valuable hands-on experience. The future belongs to the curious, embracing the journey of discovery. The field of genetics is constantly evolving, presenting endless possibilities for exploration. If you're curious about the natural world and passionate about making a difference, a career in genetics could be the perfect fit. Remember, the journey of discovery begins with a single step. Embrace the challenge, unleash your curiosity, and embark on a rewarding adventure into the captivating world of genetics. The power of genetics, shaping our understanding of the world. Our captivating exploration of genetics has taken us on a remarkable journey. We delved into the fundamental principles of inheritance, unraveled the intricate dance of DNA replication and protein synthesis, and witnessed the power of DNA technology in revolutionizing healthcare and our understanding of ourselves. As we stand at the precipice of exciting discoveries in the field of genetics, it's time to reflect on the profound impact this science has on our world. Genetics, a unifying force in biology. Genetics has become the unifying force in biology, providing a framework for understanding the diversity of life, from the simplest bacteria to complex organisms like humans. The universal language of DNA connects all living things, revealing the deep evolutionary relationships that bind us together. By studying the genetic code, we can trace the history of life on Earth, understand how organisms adapt to their environment, and even predict future evolutionary changes. The ethical landscape, navigating the power of genetics. The immense power of genetics comes with a significant responsibility. Ethical considerations surround various aspects of this field, such as gene editing. The ability to modify the human germline raises concerns about unintended consequences for future generations. Open dialogue and clear regulations are crucial to ensure responsible use of this powerful technology. Genetic privacy. Protecting an individual's genetic information is paramount. Strict regulations and public awareness are essential to prevent misuse of genetic data by employers, insurance companies, or even governments. Genetic discrimination. Genetic information should not be used to discriminate against individuals when it comes to employment, insurance, or access to healthcare. Anti-discrimination laws must be upheld and potential loopholes addressed. Building a brighter future, harnessing the power of genetics for the greater good. Despite the ethical challenges, the potential of genetics to improve human health and well-being is undeniable. Here are some ways genetics can shape a brighter future. Personalized medicine. By understanding an individual's unique genetic makeup, doctors can tailor treatment plans for diseases, leading to more effective therapies with fewer side effects. Gene therapy. Developing treatments that correct or replace faulty genes holds immense promise for curing genetic disorders and improving the lives of millions. Sustainable agriculture. Genetic engineering can help create new crop varieties that are more resistant to disease and drought, fostering food security for a growing population. Conservation efforts. Using genetic analysis, scientists can identify endangered species at risk due to inbreeding and develop strategies for their protection. The future of genetics, a tapestry yet to be unfolded. Our understanding of genetics is constantly evolving. New technologies are emerging at an unprecedented pace, offering exciting possibilities for the future. The field of epigenetics, for example, explores how environmental factors can influence gene expression without altering the DNA sequence itself. This opens doors for understanding the interplay between genes and environment in shaping our health and development. A shared responsibility. Fostering public engagement. Genetics is not just a scientific discipline, it's a force that shapes our society. Fostering public engagement with genetics is crucial for informed decision-making about emerging technologies. Science education programs need to integrate genetics into their curriculum, and scientists must actively engage with the public in clear and accessible communication. A tapestry woven in DNA. Our journey has revealed the captivating story of life, a story written in the language of DNA. Genetics has unveiled the blueprint of life, the engine of inheritance, and the intricate dance of protein synthesis. As we move forward, let us use this knowledge responsibly, embrace the ethical challenges, and harness the power of genetics to create a brighter future for all living things. The tapestry of life continues to unfold, and genetics remains at the heart of this captivating story.